Welcome to the talk show of the International Light Association. We meet twice a month live on alternate Wednesdays. And if you miss a live event, you can see the recordings of the shows on the ELA YouTube channel. We are excited to present to you experts in the field of light, color, and sound, and how these energies or frequencies can be applied to advance health and well-being. In this talk show, you learn interesting facts from experts around the world, and you will not only be able to listen to the presentations and debates, but you will also be able to ask these experts your questions. The ELA invites you to join us on this journey of learning and exploring the vast possibilities to use light, color, and sound for your well-being.
Welcome for our second show this season. We are very happy to have all of you. That was a message from our president, Thelma van der Werf. She is in New Zealand and sends greetings from there. Today we have a very exciting show. You would remember, some of you who attended the last show, and which is already on YouTube, that we had Anadi Martel, the inventor of Sensora, and Dr. Jacob Lieberman at the show 2 weeks ago. Today, we have two phenomenal speakers. The first one is Dr. Mark Rea, and the second speaker is Dr. Larry Wallace. Brief introduction to the first speaker right now. Dr. Mark Rea is a prominent vision scientist who has, how do I describe him? There are so many things that he's done over his career I would start with that he's the editor of the 8th and 9th edition of the Illuminating Engineering Society Handbook. He was the director of the Lighting Research Center from 1988 to 2017. And now he is a professor at the Department of, Population. I know new school that he's just doing, yes, Department of Population Health Science and Policy at Mount Sinai Hospital here in New York City. He is a prominent circadian photobiology scientist. Mesopic vision and psychological responses to light are two other topics that he has done serious research on. He's the author of over 250 scientific papers and technical papers. And today we are going to talk about circadian lighting. Just do it, as the title says. So Mark, welcome. It's great to have you here. Hope you're well. Hope upstate New York is treating you well. And it's good to see you.
Thank you, Abe. Can you hear me all right? Is everything good on the audio and the video? Is that okay?
Yes, you sound great.
Okay, super. Well, I want to, first of all, thank you for this invitation. Because you folks in the audience are at the tip of the spear of making a difference in people's lives. And what I hope to do is to give you some sense of comfort to do something that I think is profoundly important, and thus just do it title. I'd like for you to, before I begin, think about the Russian nesting dolls. You have, the surface one, but each time you pick away a layer, there's another more intricate, more interesting detail that you can gather from that. And the message I want to get across is that from you folks that are at the tip of that spear, you can proceed with that first nesting doll and make a difference in people's lives. And so I hope that I can answer any questions you might have on uncertainty, but I'm convinced, as many others are, that you folks can make a big difference in people's ability to sleep, depression, basically reducing sleepiness during the day and sleeping better at night, and things as type 2 diabetes and breast cancer, I think, are also implicated. So we could have the first slide. Justin, can you make that work?
There we go.
Thank you. So it's a little strange for me. I've been at RPI for 30 years, and so starting a new position at Mount Sinai in New York. It's been convenient because I can work remotely. I'm actually in Troy still today, but it's the Mount Sinai School of Medicine is noted for its innovations, and certainly I think it's the largest research hospital in the United States. So it's a real honor to be part of that team now. Next. So these will just click through, Justin, so we can dispense with that. Just go, yeah. So I want to talk about the UL 24480 design guideline, which is now a little over a year old. And we developed that, well, sorry, not so quick, thank you. So what I want to do today is tell you what it is, why it was needed, and hopefully why you should believe it as something you can do as a professional and deliver circadian light. What are the alternatives? Because there are some. And how do I get started? So I'll try to, next please, Justin. So what is UL 24480? Well, it's a design guideline for you folks. It's specifically aimed at you folks for daytime work environments that support circadian entrainment for day active and night resting people. It is not a document to fix jet lag or shift work. So it's for your 80% of the people that I'll say work 9 to 5 or thereabouts. Now, it provides something beyond good intentions. It provides a light measurement and specification methodology, and it really is specifically aimed at improving sleep. Now, what's important, and what, as Abe said in the introduction, I've edited 2 IES handbooks and have been actively involved in a number of committees. To my knowledge, it's the first recommendation or design guideline that actually holds itself accountable. We're saying if you do this, the people in those spaces will actually sleep better as you measure it by those of you that have an Apple Watch and look at your sleep function every morning. If you do, those are the kinds of outcome measures that we believe this intervention for offices and factories and schools is going to show up as better sleep. Next. So this is some examples. I think what's the science aside, the fact that you can do this implementation in submarines, intensive care units, senior residences, in your run-of-the-mill offices, including ones in Latvia and Iceland. In every case, people that are exposed to bright light during the day and dim light at night sleep better. They work in the submarines better as teams. And the performance of these people improves. So think of it, if you will, as that large Russian stacking doll. That's what this is telling you, that this actually works, and it works in a variety of environments, not just in the laboratory, but in the real world. Next. Next. So why should I believe this? Well, it isn't a hobby horse that I got a bunch of graduate students together and we wrote this up. It followed the collaborative standards development system, which all standards do. It's an orthodox procedure, very strict, has to have a scientific foundation, has to have a diverse group of experts that represent it. And you have to go through 2 rounds of public input, and you have to write written comments to everyone that writes it in. So we went through that whole process. So it is no different than any other standard. The process is no different than any other standard that you folks would follow. Next. Now, this is intended to intimidate you. This is the smallest of those Russian stacking dolls. But it has to have a scientific foundation. And what this represents is a schematic of the retina for the circuit, if you will, that converts photons on the retina to signals that go to the brain. And you can see it's complicated, but you don't need to know that. You just need to know, and it doesn't mean you can never know, or doesn't mean it's not accessible. You just have to pick a few of those stacking dolls and get down to this level, but it is there, is the key point. Next.
Hey, can you just go back to the previous
Sure.
slide? I have a quick question. And because, Mark, the audience is from all over the world, so just a few clarifications. Troy is a beautiful scenic town in upstate New York. Strongly recommend you visit it. The second is that the IES is the Illuminating Engineering Society. It's a worldwide body. And the North American one is called IES-NA. I just wanted to put that in because the audience members, are from different parts of the world.
I thought you were going to ask me about the H2 horizontal cell. That's what I thought you were going to ask.
I was going to ask you, but I thought I would just, give a little clarification. Yes. The LGN and the SCN. Can you help me with that? The bottom two?
Yeah, the LGN is the lateral geniculate nucleus, and this is your first relay station from the eye to the brain, and then it goes to the visual cortex. And there are a set of neurons that collect information from the retina and then send it down the optic nerve to the lateral geniculate nucleus. Now, close to that physically is the suprachiasmatic nuclei, SCN. And these are the ones that, this is the site of the biological clock. This is what gives us a 24-hour pattern for physiology and behavior. And the way we synchronize our physiology and behavior, this master clock, is by sunrise and sunset. And of course, since 90% of your day is spent indoors, we have to be able to emulate in some way that light-dark pattern that we've evolved, and all species on the planet evolved under, bright days and dim nights. So our challenge as professionals is to understand the science so that we can mathematically, that's what this is about, characterize those photons to give you enough signal of bright light and dim, bright light during the day and dim light at night. That's what this is all about, is the mathematics and the neurophysiology that underline that conversion of photons to neural signals that reach the biological clock in the SCN.
Great, thanks.
So the outcome of that is this rather odd spectral sensitivity function. You'll see that it's of two types. One dealing with things that are warm light sources, that's the red, and one's dealing with cool light sources, which is the blue. And so you can see here how well the model actually fits the data. And this is just an example. We've got a number of other mathematical fits to the empirical data. If you click the next one. And what you see here is if you take, you can see all the experiments that were involved. And this tells you from threshold, what is dark, what is dim. And by the way, what is dark and dim for the circadian system, you can see perfectly well. Street lighting that we have on IES, Illuminating Engineering Society recommendations, are good for vision, but the biological clock can't see it at that dim a light level, all the way to saturation, which is that upper part. And that tells you that will you be outdoors during the daytime. So all of the experiments that we've done are plotted there to show you that the mathematics is able to predict within some uncertainty the data that we've gathered in the laboratory specifically to measure that input, output, or absolute sensitivity to circadian light. Next. So the other thing, why should I believe it? This is the task group, and you can see, you may recognize some of the names here, but we had input from, NEMA, from the US Navy, from the Salk Institute, from GE, the list goes on and on of people, General Service Administration, trying to make sure that you had a diverse group of people who had a stake in the outcome of this document, UL 24480. Next. So what are the alternatives? Well, the metaphor I'm going to use is ice cream. You can go with equivalent melanopic lux as represented by the well standard, or photopic lux if you want to make it easy, because things like AGI use things in photopic illuminance. The method shows you how to do those. You're not boxed into a particular metric. We use circadian stimulus, which was that curve that I showed you with all the data. But that in of itself is not enough. That's why you add the ice cream cone. This has to be applied for 2 hours during the morning, ideally, but 2 hours during the day. So it isn't just a photometric specification. It also includes time. So think of it as a dose. You have to have the right amount and the right duration of exposure to promote circadian entrainment. So this is a call for lighting controls. Some people have said you're going to spend more energy. That's not necessarily true. You can, but you don't have to meet the design guideline for UL 24480. Next. And so how do I get started? Well, you can download this. You can purchase it, which from UL. This is the screenshot of the page that lets you do it. You can sort through it. Next, I think we just got a couple animations here. And so what I'm suggesting is that it's a pretty straightforward document, and I hope you understand that it was a thoroughly vetted recommendation. It's got a scientific foundation. Some people wonder why UL got involved, and I'd be happy to explain that if anybody's interested. But this is too late. We should have been doing this 3 or 4 years ago when we knew enough about that largest Russian stacking doll. This is something that works, and it works in every environment that we have, as long as you can deliver those prescriptions, not prescriptions, those guidelines to people in offices, schools, and factories. So happy to answer any questions if you have any.
Well, thank you, Mark. This is really great. There are a few questions that came in before the talk, and I'm sure there'll be more as we talk. Few questions of clarifications. One is when you did that diagram, maybe we can pull that up again, that looked like a circuit board.
Oh,
yeah. The connection of that with what's happening in the industry, right? There seems to be a disconnect. I mean, there's some beautiful research that you have done, right? And that your colleagues and people who have been connected with you. But when it comes down to seeing the industry at large, the profession at large, not just in architecture, but generally in vision science and talking about physiology, there seems to be a disconnect. And I've been dying to ask you this question. Where do you think is this disconnect? Why is this information not more widely available?
Well, that's why I tried to use the metaphor of the Russian stacking dolls. Most people aren't trained like I was in the biophysics of the retina, and you can't reasonably expect people to be understanding that. So what you do is you create this task group that have a variety of skills, and then you put together those perspectives. So the confidence isn't just me saying it, it's something that these people had enough time to spend to really sort of dig into it as far as they could. But the reason you have recommendations from an authority is because you are assuming that they know what they're talking about. And if you don't believe that, then that's a problem. That's not very effective. I think that we have relied a lot on the Illuminating Engineering Society as that source of authority. And I think this is just an alternative to that, but it doesn't mean one's better or worse. It just means that it's an alternative. So you get a second opinion from two physicians that are equally qualified. That's a good thing. And I think we will in the end. Just the UL one came out first, and I think that's what it is. Now, to be honest, people have livings to make, and, they probably don't want to get into all those details. So that connection that you're talking about, Abe, is going to take some work. And I think the kinds of things that you're doing right now with this kind of presentation really does help make that bridge. And I think that's what it needs. It needs to be socialized as well as, well, here's 5 scientific articles, go read them, and you'll be able to figure it out. So I think it has to be socialized like we're doing now.
Good point. Yeah, it has to be mainstream for sure. I mean, I look forward to the day when, without sounding geeky or nerdy, that circadian rhythms and how it impacts us becomes a dinner time conversation as against talking about what happened in the elections or whatever else it be, right? So, but that remains a dream of mainstreaming important research. So yeah, this is a wonderful segue to my next question, and I'm going to get to the 2 questions that have come in, and I'll get to that in just a second. But just to complete the point, what role do you think the manufacturers have in wider absorption within the market? I have an opinion on that, but I'll get to that once you share with me what you think.
Well, I don't, nothing is, I don't want to oversimplify it, but I think that most of the lighting design profession go to the manufacturers for technical information. And if they don't understand it, if they haven't gone down to those nesting dolls to the level where they can do it, they're, I don't mean to be unfair, but their primary job is to sell stuff, and they may or may not have the grounding to provide good advice. Not that they're bad people or they're lying or cheating. I don't mean that, because I think they do have good intentions. They want to do the right thing, but they may not be really qualified to give an opinion. And or they'll say, well, it's still out. Jury's still out, and we don't really know enough about the science, which I would say is nonsense. I mean, we do. And so that's why you should just do it. And I think that it is the kind of thing that professionals have to step up and make a difference, rather than rely strictly on the manufacturer's information.
For sure. And I think professionals at large have to be ready to step out of our comfort zones and try to absorb that within the work that we do. And I'm very fortunate that everybody that I know in the ILA are pioneers and innovators like that. There was a saying that I heard from somebody in one of the conferences, a prominent scientist. He says, if you're not on the Quack Watch, then you haven't really done much. Right. So I mean, that's kind of the other perspective to it. But we have we have a few interesting questions here. And I would like to get to one, which is it all about bright or dim, steady light? Or is there room for pulsing light? In that case, if there is room, that is, for pulsations, what's the role of fade in and fade outs? And is there such a thing at all?
Well, I think I'd be preaching to the choir to say that design is the integration of various objectives. Circadian lighting is one objective, but I'm not sure that anybody wants to sit in front of flashing lights all day, which doesn't mean we can't do that in the laboratory. And people have done that and shown that flashing lights can be effective for driving the clock, the circadian clock. But do you really want to do that all day? I mean, so I don't know what you mean by pulse, rapid pulses, or do you mean a 2-hour pulse, or the duration of the pulse matters, is what I'm saying.
Right. Yeah, and just to extrapolate on because I know the person who asked the question, he's talking about pulsations which are happening at a higher frequency, not at the point of like what we used to have with the magnetic ballast at 50, 60 hertz and 30 hertz, etc. But pulsating light that's used often in light therapy to trigger responses within your brain waves.
Well, again, even the smallest of those stacking dolls is not the smallest possible. I don't actually know the answer to that question. These temporal dynamics of the circadian clock are not completely, or I will say not well understood. But I don't even speculate whether it makes a difference or not. I mean, I can tell you that you integrate the energy and it can drive the clock. That's true. But whether it has other collateral issues associated with it, I don't feel really qualified to say.
Okay, fair enough. Fair enough. So there are a whole host of questions that are coming in. So I'll get to the next one here. You mentioned street light levels are not relevant yet. In a recent, I think, yeah, there's a period there. In a recent European study, melatonin suppression was recorded in a participant at 6 lux. So I guess it's a comment more than a question, but maybe you can respond to the comment here.
Well, the first question is 6 lux where? And what matters is how much light actually reaches the retina. And so the second point is that lux isn't really the right way to measure it. Lux has a bias towards long wavelengths. So 6 lux of a blue LED is a lot of light for the circadian system. And so you are looking at, I'll just make a rough equivalent, 6 lux of blue light at the cornea, at the eye, is probably equivalent to 350 lux of white light in terms of its ability to activate the circadian system. The reason we use lux is that we throw out the information on the spectral power distribution, but you can't do that and come up with a meaningful interpretation of how effective 6 lux is without knowing the weighting spectrum that we showed, or at least knowing how effective it is for the circadian system. So no question that 6 lux of blue LEDs is going to be a full driver for the circadian system. 6 lux of high pressure sodium is the system's completely blind to it. It might as well be dark for 6 lux of high pressure sodium.
Understood. And thank you for clarifying that. And the next question that's come in is, this audience member is looking for clarity on red versus blue light in AM, PM, because they're finding information that is out there to be contradictory. They've cited another research, Jacob Lieberman's book from 1990. But would you please help us understand, for those of us who are new to the concept, what kind of light is good in the morning? What kind of light what should we be doing during the course of the day?
Well, I hope my answer isn't too windy, so I'll try to be precise. As far as I know, we were one of the first labs to show that you could apply red light at night and increase people's alertness. By alertness, I mean subjectively feeling more awake, and also things like reaction time. For simple tasks, simple cognitive tasks, giving red light was an activating stimulus. We've since shown it works in the daytime. We've applied it in Istanbul train operators situation for the US Air Force. But the reason red light is used is because it has no effect on the circadian system, which is what that circuit diagram was all about. So if you want to stimulate the circadian clock, you're going to have to use, and I'll say broadly, blue light to activate it or deactivate it. Red light, it's blind to red light, but there are other pathways from the retina to the brain that affect how alert one is going to be. So we are in the midst now of a study where we're giving night shift nurses red light as sort of a red light shower, if you will. But we've already shown you don't affect melatonin or circadian phase or any of those other things, but it does have this other layer that you can apply at night in particular, so that you don't upset circadian rhythms, but you still keep people more awake. And I'm not saying don't drink coffee. I'm just saying it does add to the alertness at night. So during the daytime, the way we've done this is that we will give, and I don't want to oversimplify this. White light is as fine as blue light, but let's say bluish light in the morning for that 2 hours. That sets your clock to make sure that it knows what time of day it is, which is the morning. And then things like post-lunch dip, we apply red light, and in fact does improve performance during that post-lunch dip without having any effect whatsoever on the circadian clock. So I think that we can't think of this as some monolithic blue light impacts everything story. There are different channels for the visual system. This non-visual alerting system is also plays a significant role. So understanding that, I think, is critical. I'm not quite sure what the confusion is you're referring to in the literature, but I hope that's clear what I said anyway.
Yeah, it's basically said in that there was a book by a person well known to us and a speaker at the last talk, which is um, I don't remember this section, but what's being cited is that blue light is recommended at night and red discouraged. So that's why they were. But you answered that in terms of the circadian suppression, red has a very limited impact, and it would be through the other channels that the red is kind of working on your physiology and psychology.
So let me add, if I may. There may be other factors that I don't understand about why blue light at night might be beneficial. But I do know that blue light of the right amount and the duration will postpone the time you fall asleep. And if you have an alarm clock at 5:30 in the morning, like I do, your sleep will be robbed by having sufficient duration and amount of blue light in the evening. That's how the clock works. So I know that there may be other benefits to blue light that I'm unaware of. So I'm not throwing stones. I just my argument is sticking strictly to how light affects the biological clock.
The next question here, boy, you have a lot of questions that have come in. Would tunable indoor lighting be helpful in connection to circadian entrainment?
Well, it can, but I think that concept is oversold. The best thing you can do is sit in front of your window. And there's, I mean, you can argue that's tunable or it's not tunable, but the point is that vertical illumination at the face is what's critical. And you can do that with any white light. I mean, and you can have the same white light all day long. You just have to control the amount and the duration. And white light's perfectly fine. You don't need that. You can do it, and you may have multiple reasons to do it. For people like to see it change or any number of things. So I'm not saying that's not a good idea. All I'm saying is that the biological clock doesn't care.
So, so all of these manufacturers who come in and say that the solution to every problem that you may have is having tunable CCT, tunable color temperature light fixtures are basically exaggerating. The claims are clearly exaggerated then.
Well, it's just wrong.
Yeah. Okay, it's just plain wrong. Okay, that's even better than exaggeration. No, I mean, as a lighting specifier, I'll tell you, Mark. I mean, you've been connected with the field a long time. That the number of times that we have manufacturers who come and feed us this, and when you question them, the responses is, but everyone's doing it. Well, that doesn't mean it's right. That doesn't mean it's accurate. So I thank you for your clarification there. And I think we have time for, how are we doing? Two more questions, but there's one that's come in, which should be a simple, connection to a resource, which is, do we have any table showing the percentage of melatonin suppression and the corresponding light level CCT and SPD? I'm guessing the well standards have them too, but please, Mark, if you would like to answer.
That's all in the EWEL 24480. But you can also look at the LRC website and make those conversions. We have a calculator that's free online, and you can put in any spectral power distribution. You can even have 20% daylight and 40% warm LED, and you can do the calculations that way as well. So all of that is readily available by the that free website that we have. It's called, I think it's CS Calculator. If you do LR's Lighting Research Center CS Calculator, it will take you right to it.
I put the link in the chat. It's lrc.rpi.edu. And you can find the CS calculator there. That'll help you with the SPD, CCT, etc. There's one more question, which is a comment, not a question. Thank you for this discussion on red-blue lighting. I was just looking at the alternating red light, blue light training device from iPod. And there's studies that it increased batting accuracy on a little league, so that'll be baseball, by 90%. I like the idea of being more accurate in anything, and it corrects astigmatism. Kind of pricey though. Well, thank you for sharing. And those of you who wish to look at this, please look at the comment box. It's a comment from Trisha. Very interesting. Do you want to comment on that, or?
No, I'm out of my depth on that one. I can't comment. I mean, I like to talk, but I don't know much about it, so I felt it would be a disservice for me to give an opinion on that at all.
Okay. Well, yeah, you probably have to do www. Just to Lighting Research Center. It should show up as a link. I just got a message there. All right, so you said, why did UL get involved? Right? You said that, that somebody may want to know. I would love to know why, and I think a few audience members also would, that why would UL get involved in this?
Well, I think I heard a prominent lighting designer say that UL should just stay in its lane, shouldn't do this. But the truth is that nothing was happening.
Somebody I know as well, or probably.
Well, I'm not going to mention any names, but the point is that
yes.
The Lighting Research Center has always been in that position that when you step up, we have. I don't know if you remember polarized luminaires and
sure, yeah,
electronic ballast and why are they failing, and
yeah. The end of life stuff. Yeah, I remember that.
One of the recent ones was the AMA thing about light at night and 3000K versus 4000K. I mean, we feel obliged to step forward. I mean, as an academic, if you don't do that, you're really violating the implicit part of tenure, which is that you should be able to speak your mind and not be afraid of losing your job, which isn't true for many, many people. So the LRC worked with UL, which was an unusual thing to do. But, when somebody says stay in your lane, it's interesting. It's not a matter of right and wrong. It's not, like, well, you screwed it up or you didn't follow the procedure and stuff. It's just like, well, you're not in that tribe. And that doesn't seem like a good reason, certainly not one for the Lighting Research Center to back away from. So that's why we did it. And the IS still doesn't have anything out, right? And ours has been out for a year. So we started after they did, and finished before they did. But that's, again, what the LRC does. So.
Yeah, that's for sure. Well, one last question, Mark, before we let you go, and thank you for your time and patience with all of these questions. I would love to know, what is your next big project or your dream project, the project that you've always wanted to do. Now you've moved, there's been a transition from the Lighting Research Center over to Mount Sinai. What do you hope to achieve in your research now?
Well, I think the new area for us is ultraviolet, and I don't mean just for germicidal infections, but I believe there's so much potential for this audience as well to begin to embrace ultraviolet as another dimension of effective lighting design. And so I think pioneering what works and what doesn't. We just published a report, which I hope many of you can go to the website and see what we've done. But there's a lot of uncertainty out there about what do you do about ultraviolet, I put it. And I hope this report reduces your uncertainty. There's still a lot of unknowns, but I think buying hand wands and waving them over the bedspread is something you probably aren't going to want to do. And taking UV and put it on a bus is probably you don't want to do. But there was a lot of things, some innovations coming along that I think are really exciting. And because of LEDs, we aren't stuck with just 254 nanometers anymore. So I would say there's a quarter century of really interesting work to do on ultraviolet. And of course, we're doing it to treat plant diseases, avoiding chemistry to treat them. It's more effective, more cost effective. We have a couple innovations that I think could change the way agricultural treatment. But UV is, it sounds like The Graduate, like plastics. I think UV is going to be the future for a lot of people in, I'll call it lighting, even though it's strictly not light. I think that's where we need to go. And certainly in a healthcare environment, getting a good handle on UV is, and building on all the work that's been done for half a century, I think is really what's excited me right now.
Fantastic. Thank you, Mark. This is really informative, and I'm very thankful for your time, for coming to meet us here at the
I'm honored. Like I said, you folks are at the tip of the spear. You're the ones that can improve society, and I like for you to think of it as the largest of the Russian stackable dolls. It's something you can do today, and I hope you will. I do.
Thank you, and I hope you would involve with what we are doing at the International Light Association, and hope you will visit us again soon.
Well, you're very kind. Thank you. Thanks for asking.
Thank you, Mark. Thanks for being here. All right, so we are moving on. Thanks, Mark. We are moving on to our next speaker, Dr. Larry Wallace, based in Ithaca, New York. He's the former president and current education director at the College of Syntonic Optometry. He's the past president of the ILA, the inventor of the electrostim device, which is a bioelectronic device for treating macular degeneration. He's a patent holder on the first microcurrent device to treat retinal diseases. He has held numerous workshops, given numerous lectures all over the world over the past many decades. In fact, the first time I got introduced to the ILA was when I met Larry at the conference in Vienna, and he told me more about it, and I ended up becoming a member and sticking around. That was 5 years ago. And Larry, it's wonderful to have you here. I'm really looking forward to your talk today on ocular phototherapy. I'm sure there are a lot of audience members here who will have a lot of questions for you. There are a few that we couldn't answer in Mark's. Maybe we can get that in towards the end of your talk. So welcome, Larry. Thanks for taking the time. It's good to have you here.
Yeah, thank you, Ape, very much. Thank you for having me.
Okay.
All right, so we're going to talk about ocular phototherapy, and that's going to be more of a therapeutic application of light. And it's going to be involving mostly neurology and physiology, because that's the basis of our exploration of how to therapeutically apply light. So Justin, if you could put on the first slide, thank you. Okay, so the first part of this presentation, we're going to talk about syntonics, which is the application of visible light into the eye to treat a whole host of visual problems. Dr. Riley Spittler, who wrote the syntonic principle in 1941, was a physician. He was also an optometrist, a physical therapist, and a naturopath. But his real love was for optometry, and he did this thesis, and which evolved into the syntonic principle. And that principle was explored by all kinds of research on both people and animals and plants, and research of the day based on biophysics, on neurology, psychology, and physiology. So it had a lot of scientific basis to the evolving of this principle. Next slide, please. So we could define syntonic phototherapy as the application of specific light frequencies, that is in the visible spectrum, into the eyes to restore syntony or balance in the autonomic nervous system, which is crucial in supporting vision. So syntonics is really aimed at restoring of a balance in the autonomic by affecting changes in the sympathetic and parasympathetic systems. Imbalances in those systems create vision problems that often can be addressed by treating them at the source, which is in the brain, with light. Next slide, please. So he did his research based on the effect of light frequencies, and frequency means not the color, the subjective color, but the actual frequency and the energy content of the light itself, affecting our bodily health, our inherent electrical and energy systems. That is, he looked at the body as in physiology as electromagnetic charge. The body was actually like a cell, like a battery cell that has charges that drive most of all of our biochemical reactions. He looked at the physiology of the eye in terms of application for ocular pathology, but especially for ocular function, and that means things like eye coordination, eye movement skills, focusing skills, peripheral vision, and visual processing of light information. And that light itself affects the emotional centers in our brain and the emotions that we express. Next slide, please. So that he evolved 21 principles, which we're not going to go through, but each one elicits or elucidates the specific effect of light on various physical and biological systems in our body. And he said that it was the select application of visible light that you could use to measure, or not to measure, but to affect changes in our sensory motor systems, especially ocular motor, both sensory motor, the endocrine system through the pituitary gland, pineal gland, and the hypothalamus. Next slide. So this system uses 11 frequencies of light, which are light combinations of different filters to treat binocular and sensory motor conditions. This outcome of this treatment affects visually related attention and memory disorders, accommodation and convergence problems, ocular pathology within physiological limits, asthenopia, that's eye strain and headaches, special application for closed head injuries. And what we see its effect most profoundly in vision is expansion of the visual fields, which were often constricted and have defects as a result of many physiological imbalances that are imposed on our systems. Next slide. So syntonic phototherapy affects the physical. That is, it has effects on our bones, on our organs, our tissues, and our cells. And these are all explored in the literature. It has chemical effects, and those are primarily through the neuroendocrine system, our hormones, and things like ATP production. It's energy production within the cells and within the whole physical system. It has physiological effects on our nerves, on the energy within our body, and therefore our metabolism. And light has specific effects on our psychological emotions and, of course, our mind. Light is entering our eyes through the pupil. This is a schematic or a picture of a pupil. The pupil's the most sophisticated electronic scanner that you can imagine. It's changing its size, in its diameter and its constriction constantly, thousands of times a day, to modulate the amount of light entering our eyes into our brain. The pupil is one of the two major ways we can monitor the autonomic nervous system in real time. As you can see what's affecting the autonomic system, sympathetic, parasympathetic, balancing, and how they interact with each other by changes moment to moment in the pupils. So we use pupil diagnosis as one of the major criteria to see if our therapy is working. We'll see abnormal pupil responses proportionate to the imbalances in our nervous system and even how our visual system is operating. And as we do therapy, the pupil measurements will actually change, and we can see how we're affecting the autonomic system again in real time. The only other way which we have right now to measure the autonomics in real time is heart rate variability, which also shows the interaction of the sympathetic parasympathetic autonomics. Next slide, please. So as Mark was saying, there are whole other channels that besides going to the cortex of the brain for vision, there are non-image forming visual connections, and he was talking specifically about the LGN and SCN. In Syntonix, we're interested in a number of different pathways. Two of the most important is the thalamus and the hypothalamus. There are direct connections to the thalamus, in the hypothalamus in the midbrain, and also to the pituitary gland. In addition, there's connections directly and interacting through cybernetic feedback systems, also with the superior colliculus and the vestibular system and the brainstem. So there's all these networks that are not have to do with vision imaging, but have to do with where the energy, this whole non-optic track, it's called the retinohypothalamic pathway, is sending energy to all various kinds of brain centers. And through the application of light frequency, you can actually change the way these, the frequency distribution of energy is going to these different brain centers. Next slide. So the thalamus simply is a gating system for attention and arousal. It has electrical discharges, and its light frequency can set points to stimulate or inhibit the system. So what this means is all the sensory input coming into our system and all the motor output going out is all going through the thalamus, is modulated by the activity of frequency distribution of light energy through the thalamus. And the thalamus is specifically can be set, reset by using specific frequencies of light to change the way the sensory motor system is working. That is, you can increase sensory input, increase motor output, or vice versa. Next slide, please. And then the hypothalamus is the other area that's so critical, direct input, and the hypothalamus is really controlling so much of our unconscious inner regulation of our nervous system, our autonomics. And that includes the digestive system, GI tract, metabolism, respiration, breath rate, heart rate, emotions, sex drive, body temperature, sleep cycles, and immune function. And this hypothalamus is continuous with the pituitary gland, and also with the third ventricle of the brain and the diencephalon, which is the midbrain. And this continuation of light energy going through these different pathways can modify all these different aspects of our physiology. Yes, next. When we shine light into the eyes, the picture of the retina, when we look through your pupil, you can see the back of the eye, where the disc is, this round picture here. This is where the optic nerve is, and this is the retina. And this, in the middle here is a place called the macula, where the central acuity is, and all the rest of the retina is peripheral vision. And when we shine light in the eye, we're actually irradiating the blood supply to the eye and also to the whole body. It's been shown that 40% of our blood supply of our body moves through the eye, through the retina. And next slide too, please. Through the retina and also through the choroid, which underlies the retina. About 40 to maybe even more of our blood, but at least 40% of our blood supply goes through the eye every 20 minutes. And that's the duration of a treatment using syntonic light therapy. So we are radiating the blood. The blood is full of chromoacceptors, hemoglobin, all kinds of other actually light absorbing molecules that then signal all kinds of physiological effects throughout the system also. So not only is there a neuro pathways, but there's also by circulatory system is also sending light messaging throughout our whole body. Next slide, please. So the other
So I'm
way.
sorry, I just want to jump in very quickly.
That's okay.
So 40% of the blood supply goes through the eye in 20 minutes.
20 minutes, correct.
So if I waited another 20 minutes, would it be like 80% or is it?
Well, that would be true, but you wouldn't need to have the physiological effects. In 20 minutes, you're going to get the major photo accepting. That's like the peak duration for accepting photons into these different blood molecules.
And is it
What I'm going to get saying is that more isn't better necessarily.
more is not better, you're saying?
Doing it for 40 minutes doesn't give you twice as much effect or anything like that.
Okay, understood. Thank you, Larry.
Next slide, please. So that's just a picture of the blood supply. And so when we're putting light energy into the eye, we can direct light energy, photon energy, chemically, neurologically through the blood supply all over the whole body very rapidly. And there's no other source of photon energy into our body that's more effective than the light. The skin above the layer of the skin takes in a lot of light as well, but we're covered up most of the time. So anyway, the light is the major transducer of this kind of photon energy. Next slide, please. That's a picture of a syntonizer. That's a device where you look in for these 20-minute treatment. It has a frosted lens to diffuse the light. We put glass filters right in front of this light source. The light source is very important. We use incandescent light, which has a broader spectrum. It has infrared included. And to increase the energy output of an incandescent bulb, we use what's called a vibration bulb, where the filament vibrates. We use a transformer to upgrade the voltage from 110 to 145 to more rapidly vibrate the filament, which gives a broader spectrum, a more energetic transmission of light through this device. And you sit at the end of this tube, and this is a treatment. And that's, we'll go into that more in just a moment. Next slide, please. We're very interested in the peripheral vision. That's beside the pupil reflexes. Syntonic optometry pays especially a lot of attention to how we see peripherally. When we measure a visual field, we have to measure it not by just light detection, but we measure it how you process movement and motion. It's called the magnocellular pathway, that it's the part of the retina that doesn't see detail, but it picks up motion and it picks up low frequency light stimulation. And this visual field changes all the time for us, but when it gets reduced, like this picture here, the light input from the eye to the rest of the physiology is greatly reduced. And in treatment, we give light treatment to restore the sensitivity in the field, and we'll see the field starting to open up into this third picture, and you start to see something called the blind spot, and that's where the optic nerve is. There's no vision in that area, but the blind spot also becomes enlarged through trauma and through all kinds of different physiological imbalances. Next slide, please. So in order to prescribe light, one of the methodologies, and it's very complex to really analyze cases, but a very simple way to get people to begin to use light therapeutically through the eye is we use a syndrome analysis. And syndrome means we look at a sequence or sequela of basically symptoms and signs and symptoms and history, those three things: medical history, the signs or symptoms they may have, and then and that gives us a guide what kind of light we want to kind of prescribe. So for instance, in this lazy eye syndrome, we want to use red light, red-orange light, which is a sympathetic stimulant. We use it for lazy eye. In chronic cases where there's a lot of physiological imbalance that's related to neuroendocrine imbalances, toxicity, things like allergies, that are long-standing kinds of problems, we use yellow-green light, and that's called for the chronic kinds of problems we'll see. And we see this all reflected in the visual findings, as well as the systemic findings and the medical findings when you take a history. The acute syndrome is the use of blue-green light, which acts as a kind of a mild parasympathetic activator. And that is used for head injury and for when there's a history of high fever, when there's inflammation, anoxia, that is lack of oxygen supply to the brain, swelling, headache, double vision. When there's pain, we can add more parasympathetic light stimulation by going to indigo, which is a pain reliever, and that's a color that we'd use when there's more headache, when the eyes are hurting, when the eyes drift apart. We can stimulate their reflexes to turn the eyes inward. And another one that's commonly used a lot, of course, is for emotional fatigue, and that's when we use a ruby color or a magenta color also. It's when we put red and indigo together. It's for coping skills, mood swings, alpha mega pupil, poor pupil reflexes, but also mostly for adrenal fatigue and fatigue in the general system and emotional stress and imbalance. And so that particular frequency seems to affect in the thalamus, the emotional centers, and you can help balance emotional affect and emotional well-being through using that color. And so we use that a lot with the other colors as well.
Fantastic. Hey, Larry,
Yes.
I have a quick question. When I attended the Syntonic Optometry Conference, I understood how the alpha, delta, mu delta, upsilon, etc. terminology works, but would you share the parts of our audience that are not familiar with this annotation system?
Yeah, that system was devised because color, we didn't want to use the term of color because that's too subjective. So the College of Syntonics prescribed their specific frequencies that were developed by using Greek letters. And so Greek letters like mu delta was green, and mu was green and delta was yellow. So the Greek symbols are used to specify specific frequency output of the filters that are used. And these are glass filters, and they've been used for now, well, we're over 87 years. These filters have been applied clinically. So this has been around a long time, and Syntonics has been again, using those Greek symbols as a way of defining what the frequencies are.
And when Dinshaw was doing his work, he did not use these symbols, did he? He just used his colors that he had.
That's correct. Right.
Got it.
Right. So but his interestingly, Syntonics, I think, took a lot of information from Dinshaw, and some of the filters I think that he used were analyzed and actually probably incorporated into Syntonics as well.
Fantastic.
He was really one of the major pioneers of developing the specific, frequencies used. Although he was tonating them all in the body, and we were putting them right into the eyes.
Thanks.
Next slide, please. So I mentioned the visual field. Here's a case where a child came in, and we brought a target in, and moving in slowly, and they couldn't see it till it came way into here. And when this happened, this child's visual system was not operating well at all. In fact, the child had hyperactivity, had aggressive behavior, couldn't read, was having all kinds of problems at school, and was having actually acting out in school, and also had the habit of trying to bang her head all the time to kind of increase stimulation to her vision, which we believe, but she was a head banger as well. And so she was prescribed light, and just two lights, primarily the ruby light, the alpha omega, the red ruby for emotions, and the yellow green for chronic problems. And next slide, please. And as her field expanded, this is now a normal field, all of her behavior changed. And in this time span that she did this light therapy, her visual findings all became normal. Her hyperactivity was gone. She was now almost at grade level for reading. She was not acting out in class, and she was even in gymnastics with tremendous enthusiasm. She could now use her body in space where she couldn't before. And the parents said the biggest thing is she became a loving child. And this was all as a result of using light, opening the fields, and seeing this kind of change that correlated with, as the vision field opened, all kinds of other things changed in her whole physical, mental, and emotional health. Next slide, please. So that's just a real brief introduction to using Syntonics in that way. Another way that we're using ocular phototherapy is what we call focal syntonics. It's the application of specific colors right on the insertion point of the intraocular muscles that surround your eye. There are 6 muscles basically feeding into 3 cranial nerves that can be accessed by putting color right in around the eye. And when we do that, we can measure the range of movement of the eye. We can see if the eye cannot move in certain directions because the muscle system is tight, or it's not lacking oxygen, or fatigued, or various reasons. We can restore the energy to those muscles, and you'll see a normalization of the range of movement in the eyes. And that helps eye coordination, eye movement, and other visual functions. But also, when we did this therapy, we work with also the body. We have physical therapists doing part of our therapy. We saw shifts in the cranial bones and the dura mater that goes from the occipital bones down to the base of the spine. So we were finding that when we put light into the eye muscles, we were actually doing cranial sacral therapy via the eyes. And we were seeing, as a result of that, people's posture shift, and we saw restoration in their balance system. And that's one of the critical things we work with, especially with head injury. And there, of course, you can use, I can see somebody wrote a Molamed. Molamed is a great light source. That company it makes great lights. In fact, right now, the next picture you'll see, let's do the next slide, is a Molamed handheld lights putting into the, here it says SO and SR, that means superior oblique muscle and superior rectus muscle. Either we were going to stimulate one or relax the other in order to restore ranges of motion. Of these eye muscles are always working in conjunction with each other. One has to move one way, and then another muscle will have to go with it from the other eye. Anyway, so we'll use these kind of devices to stimulate these points.
Is this connected to Chiprikov's work of lateral chroma?
I don't know. Interesting, yeah. We'll have to talk about that. Next slide, please. So this is another device that we use. It's called we use low-level light therapy. We use cool lasers. This is something called multi-radiance application from called coherent. Well, let's go to the next slide. Let me show you what it is more. COMRA, which is the coherent multi-radiance application of light. And it's called the Delta laser, who's made by this company, COMRA. And it has 4 basic coherent energy applications. One is that it presents in a geometrical presentation of red, yellow light, violet light, and blue, green light through LEDs. And it has a certain geometrical presentation and a certain pulsation directed into the point of where you're treating. And we were, in this case, treating the eye, although they treat over 200 physical conditions with this device. It also had pulsed infrared light in the center. The pulsed infrared light was to increase ATP production in the cells that are treated. In this case, we put it in the head and the eyes. And it's also used with magnetic fields that potentiate the ATP production. When you add magnetic fields to the pulsed infrared, the ATP production goes up 200 to 300%. So that's incorporated in this one device. And it also has ultrasound. So the ultrasound is pulsed to basically increase enzyme production and increase the metabolic kind of coherence of the cells and of the areas that are being treated. So this device has been really wonderful to use with head injuries, with stroke victims at the site of where the stroke is, and to treat various kinds of paralysis in the eye muscle. So it has a lot of neurological applications in our office. Next slide, please.
This is the device from, sorry, can you go back to it? The COMRA? That's the device from those folks in South Africa, right?
No, they're actually from South America, from Chile. So yeah, some of you I'm sure know the source. I can't think of it at the moment.
Fascinating. Okay, thank you.
Yeah. Okay, and so this is just an example. I'm going to go through the findings, but for instance, we have we see people who have Parkinson's. We use that device on their head and on their eyes as a routine treatment. And in this case, the person couldn't read, their eyes were going way apart, they couldn't see very clearly, it was blurry far away, they were seeing double. We gave them a few treatments with this low-level device. The developable vision was gone, the eye coordination was normalized, their acuity went up to normal at near, contrast increased, and now the person was actually looking forward to the book sale because they could read with clarity and comfort. So this application in a condition like Parkinson's is not a curative, it's a therapeutic thing, and you have to keep doing it on a regular basis to maintain the effects. Next slide, please. And this is another light application that we use in the office for treatment. It's called a photon stimulator. It comes out of a company called Light Forms in Tucson, Arizona. It's basically a light box that you can put filters into, and it has a fiber optic system to apply light to acupuncture points and to affect microsystems. This device was used initially primarily on the ear for auricular therapy, and you can put filters into it there was about 20 different colors you can put in and deliver it through this fiber optic system. And so you could focalize the light into a very small target area. Another slide, please. And in this application, one that we use a lot is in macular degeneration. It was based on a treatment protocol by a man named Ivanek from Germany, that we would put red light focalized for 40 seconds right on the macula. We put it through the temporal part of the conjunctiva, just outside the pupil or the iris, to shine it on the macula. And we found amazing acuity improvements, sometimes 1 to 4 lines of acuity in 6 patients initially treated. One went from 2800 acuity to 2200 to 2000 to 2060.
Okay.
These improvements were lasting up to a month, and then the treatment had to be repeated. And so we've had some patients hold the improvements up to 4 months. But people have a condition like macular degeneration, we would have them purchase this device and treat themselves at home over a long period of time.
Which device was this?
This is the one I just showed, the one right before, the slide before.
The one from Tucson?
The photon stimulator, right. So we could put the small beam of light right into the macular area. We also using it for other things like glaucoma and other conditions, optic nerve problems as well. So that was my 30 minutes. I've got through my slides and gave you an overview, taking the light systemically through the eye to affect all these physiological systems, which has been an ongoing research project. One of the things we want to learn is how can light being put into these brain centers affect so many different things, and how can we refine the therapeutic application. And that's where the research is. So that's our dream, to get more research in this way. We have clinical research that shows efficacy. We have studies to show that when we do these treatments, all these things change in their vision. But understanding it is harder, because in the field of photobiomodulation, when they're using lasers, they're putting it right on cells. They know what the dosage is. They can see the changes in the cell function, the mitochondrial change, the ATP production, phosphooxidation. We can measure chemical changes on a local area, but we'll put it in the brain. We change all kinds of things in the brain. And we know that from all the therapies that are involved in the International Light Association. And so this physiological aspect is, of course, what I'm interested in as a researcher and practitioner in this field. And we want to understand the biology and physiology. And we have a lot of information. It just keeps growing like crazy all the time.
What you're saying is that we need more tests, more field statistics that we can.
Not so much that. We need more. We really need. One of the simplest ways is start to measure using functional MRIs, PET scans, SPECT scans, see what's changing in the brain. More physiological measurements. So tests in that regard, but also understanding the magnitude of how light affects us is huge. As we know in the field of biophotons, we take in light energy. It's postulated that the DNA is taking in biophotons from all the things in our environment. sending light back out and connecting us with the whole field around us, that there's fields of light energy around us, which we have now devices to measure those fields around us. We can count biophotons in our body, out of body. So there's a whole integration of all of our structure and our matter in related to light. And so the magnitude of understanding all that is gigantic, but thrilling, thrilling at the same time.
That's fantastic. That's fantastic. Thank you so much for sharing that. There are a host of questions that have come in, and a few of them came to me directly. So let me start with, in no particular order, we'll try to get to as many as we can. Is age a factor in phototherapy?
No, we treat babies, we treat infants of very, very young age, even a month or two old, up to people that are 100. The efficacy of changing the nervous system definitely diminishes with age, because we have all these comorbidities, all these chronic kinds of imbalances that are harder to alter as we get older. So we see the most amazing changes in kids, because their systems they don't have embedded health problems, but yet the application of the light can be done all through life. And we've treated people 100 years old.
Okay, thank you. And are there any contraindications to using light into the eyes?
No, there has not been any. And there's been the only thing is you can have an aggravation at times. You can be just like a physical or emotional aggravation that's very short, very limited in duration, as certain colors can be more disturbing to people. But no, there's no contraindications. In fact, it seems very evident that we taking in light all the time, and that if it's the frequency that we don't need, we just don't absorb it. And so it doesn't have a negative outcome.
Great.
It's the wonder of using light. You don't get side effects.
Right. So the next one, I'm trying to, because there are so many questions coming in, and then just making sure that I don't miss anything, because they're all wonderful questions. The next one is it a particular wavelength that causes the effect, or is it?
It's the frequency. So it's the vibration. So everything, all the transmission, all the communication between the vast network of our physiology, there's all kinds of networks. They're all communicating via frequency. If we talk about the lights that we're using, long wavelengths have lower frequency, and they also have lower energy. So that's the other component. The higher the frequency of light, such as blue, the higher, the shorter the wavelength, the higher the frequency, the higher the energy output. So we are dealing with energy, and energy through vibration. So there's both vibration and there's also energy.
That's fantastic.
But not wavelength per se.
So great. I would like to talk more about each of these, but there are so many questions, like I said, that are coming from the audience, that maybe we can chip away at them. And then if people have if they need more information, maybe they can be in touch with you, and even for Mark, directly over email, which we're happy to share with them, if that's okay with you.
Sure.
Yeah. Okay, great. So the next question is, which is kind of connected to the dream project question that I asked, but here it's asked of an audience, by an audience member, that given the new applications for color light frequencies emerging all the time, what has Larry seen recently that he wants to explore more in the future?
Well, I think that I really looking to employ more modalities, like sound. And in fact, the ILA is now moving to incorporate sound more. In fact, as we gave light treatment in the office, we were also using tuning forks that had the same octave in sound as the light. So we could actually potentiate or act synergistically with light and sound. So I think more modalities is certainly an area that, and how they interact, is very interesting. Yeah, I can go on about that.
Yeah, and in fact, since you mentioned sound, last week we had color and perception. This week we had the eye in its different ways through circadian and ocular phototherapy. Next week we actually have sound. and one professional who treats people with sound therapy, sound vibration, and she's also the head of the International Association of Sound Therapists, right?
Great.
And then we also have a musician who works with sound and who has been researching music from our ancestors. So I just thought I would, for the next show, they can stay tuned, and thank you for saying that, Larry, because I think it all kind of comes together, like you said, the tuning fork, etc. There's one statement here. I have so many questions. I would like his email, please. We'll get you that frequency. Okay. And then there was one which was about, oh Lord, 1 second. Please bear with me. Okay. If the tension of the oculomotor muscle is the main factor of myopia, what is the biggest culprit? That's
Well,
which one of them is the biggest culprit? That's the question. I'm not sure
well,
I follow it
well,
completely.
if you focalize it to the eye muscles, which is really just one aspect of it all. There's many more aspects, but it would be the oblique muscles, superior and inferior oblique, tend to squeeze the eye, elongate the eye. But you have to look at what's the source of why this is happening. Is it cranial? Is it autonomic? Is it, there's so many, is structure of the eye weakened where it loses its elasticity, or it can't stretch. I mean, there's all kinds of components.
Super. Next question. Since everything is now understood to be frequency, is there a possibility that these frequencies could be remotely transmitted?
Well, I mean, historically they have been. I mean, there's been lots of fields of radionics and other types of technologies that have been using transmitted frequencies over long distances. But that's beyond the realm of, where I would be working. But absolutely, yeah, there's all kinds of questions to that. But yes, there's certainly been lots of disciplines that have been used to transmit frequencies over long distances and have biological effects.
Okay, and this, I think we'll keep this as the last question. It's quite fantastic. And for those of you whose questions I couldn't get to, we will get you Larry's email in the chat box here. It's Larry. Well, I'll write it down also, but it's larry.colordoctor@gmail.com. Color spelled the American way, COLOR.
Yeah, Dr. Color, DOCTOR. Right.
Yeah. So please send in your questions to him. And the last question, which actually I find to be interesting, and I would love to be part of this as well, is that it says that I would love if the Syntonics College, which you're part of, and with Rea Gottlieb, I think he's in the audience today, which is fantastic. That I would love it if the Syntonics College would offer a more layperson training, or even one for natural vision educators versus the doctor-directed training they normally do, which is also good. I mean, they're all such smart people, but for an architect, lighting designer, and for the person who's asking the question, I follow it, but in terms of utilizing it on a daily basis in life, being a layperson, quote unquote, what could the Syntonics Optometry College, what are they for people like us?
Well, not at present, but they did. We had, at one point, we used to have a non-optometric track taught by psychologists and other people for non-optometrists. And that track would be going concurrently with the track for the optometrists. And we lost our faculty to do that over the years. And but it was something that we did, and we were very interested in. In our conferences, we've always brought in lots of different practitioners from all different fields, interdisciplinary fields, researchers, therapists, doctors, all different kinds of people to try to broaden that our knowledge and share scientific information. So at present, the focus has been, we've had a huge growth spurt and interest in this in our field, and our focus has been more training optometrists. So we've been doing lots of webinars and more people attending than ever. But I think it would be very great to go back and offer our thing to have that non-optometric track again.
That'd be fantastic. That'd be fantastic. And Somebody else asked earlier that they would like more information on licensed practitioners so that they could reach out to them in their area. And then somebody in the audience responded that that list is available on the website of Syntonic Optometry.
Yeah, that's csovision.org. It's College of Syntonic Optometry, csovision.org. Yeah, there it is. Great.
Yeah, csovision.org. Thank you. Super. So that's all we have time for. Larry, it's a absolute pleasure to be in the same room as you, to learn from you, for you to share your wisdom and knowledge with all of us. Deeply obliged and very grateful. Thank you for coming. Thank you for
Yes, thank you for having me. Thank all of you for attending and having interest. Yes, thank you so much.
Another round of thanks to Mark. Mark, thank you for a great talk. And those of you who wish to be in touch with him, would it be okay for them?
Sure.
Yeah.
Yeah.
So Larry's is Larry, just help me with this. I know you changed the Larry Dr. Color or Color Doc?
It's a Dr. Color.
Dr. Color at Gmail.
DOR Color at gmail.com.
And Mark's is Mark, you have a new one as well as mark.ray at mountsinai.org. Mark, that's Mount Sinai. .org. Perfect. So that's all we have time for. Thank you all of you for coming. Thank you all of you for your many questions. It was a fantastic session. And again, thank you to the speakers. Just a few housekeeping notes. The next talk will be on February 10th. As I mentioned earlier, it would be focused on vibrational therapies. Please remember to subscribe to our YouTube channel of the ILA. This recording will be available exactly 1 week from today. So you can see it on our YouTube channel. Please remember to subscribe. Please also consider becoming members of the ILA so that you could join our tribe of like-minded professionals. And for all that information, you can visit the International Light Association website that was on the invite and the light letter that you have been receiving. I would like to thank our channel partners, Colognostics, Aqua Quinta, and Soul Studio. And to our team that has helped me produce this, which is Lynn Dool in Australia, Pascal Vidal in France, Anoop Kushwaha in New Delhi, and Justin Mensch here in New York City. So thank you all for making this a great show today. And we see you in 2 weeks. Bye for now.